will for the first presentation. I would now like to introduce my co-chair, Marti, who is leading a group in Glasgow on the biology of gametocytes, Plasmodium falciparum, also other species. And today he's going to talk about a type of transmission blocking immunity that has been hinted upon in earlier work, but is so far not rigorously examined: immunity against circulating gametocytes and their implications and potential use as vaccine candidates. Matt.
Thanks, Tom. So, yes, I would like to present some data from an ongoing study that we have been doing in close collaboration with Ton Bosma and Terry Taylor and others. That is unpublished so far, where we study the targets of naturally acquired immunity against P. falciparum gametocytes. So essentially the question was, are there immunogenic proteins on the gametocyte infected red blood cell surface that could serve as novel transmission blocking vaccine candidates? And the idea behind that was that the current transmission blocking vaccine approaches have issues with inoculum size, because of course a mosquito only takes up a very small volume, so very high antibody titers are required, with the absence of natural boosting for many of these antigens, and with the issues about the readout in mosquitoes. So what would these antibodies target? A series of studies have recently been establishing a model where the major site of gametocyte development is in the human bone marrow, and actually a subset or a fraction of asexual parasites seem to be associated with that site as well. So antibodies could block the development of parasites in the bone marrow, or they could induce clearance by immune cells, for example, through phagocytosis, complement fixation, or cytotoxic killing. So far, there have been conflicting data on gametocyte surface antigens and their immune recognition. Several studies have shown recognition of mature gametocyte-infected red blood cells. So on the left, you can see a study by Colin Sutherland's group, where they used first a nucleodide to detect the infected red blood cell, here asexuals, here mature gametocytes, and then human IgG to detect surface recognition. And you can see the surface recognition of mature gametocytes and, of course, for asexual parasites. There was no detection of immature gametocytes in that study. On the other hand, Pietro Alano's group, here a paper shown on the right, has demonstrated that, for example, human bone marrow-derived mesenchymal stem cells, they can bind immature gametocytes but not mature gametocytes, suggesting that immature gametocytes have surface ligands on the red blood cell. So that was the starting point for our study, and initially we further analyzed the data set that Will Stone just presented, where 3 cohorts have been, the sera from 3 cohorts of malaria patients have been analyzed with this protein array that was developed in collaboration with Phil Felton's group. So what we did essentially was to stratify the proteins on this array by stage expression. So there is proteins that are expressed in asexual and gametocytes, these ones here, or only in asexual stages, or only in gametocytes, or in gametocytes and mosquito stages, or only in mosquito stages. When you then further stratified by whether these proteins are exported into the host cell and even the surface, or on the merozoite or gamete surface, or whether they were parasite internal, we got some very interesting, significantly differential data. You can see in shared antigens and asexual antigens, the secreted antigens mount a much stronger immune response than the parasite internal ones. That's also true for gametocyte-specific and gametocyte and mosquito-specific antigens, but the response is weaker. Overall, we also saw for many of these secreted antigens responses that increased with age, suggesting some exposure-dependent accumulation of IgG. So then, when we go back to these flow cytometry assays that have been performed before by other groups, we looked at stage 2 gametocytes and stage 5 gametocytes. This was now in a cohort of 244 patients in a cross-sectional cohort from Malawi. And you can see gating for infected red blood cells with a nucleodide. Taking that population shows a fraction of cells that are positive, these are live cells, labeled on the surface with human IgG. In this case, about 10% of stage 2 gametocytes. With stage 5 then, if we again gate first for infected red blood cells, then we exclude all the spontaneously activated gametocytes using 48-45 antibodies, there's very little labeling, about 1% of the cells. When we do the opposite and exclude all those that are intact red blood cells with the glycophorin C antibody, then you have about 50% reactivity. So essentially that suggests that the signal coming from mature gametocyte-infected red blood cells is mostly spontaneously activated gametes, or gametocytes and gamete recognition. Overall, in this study, we found that an average of about 10%, 8 to 10% of stage 2 gametocytes are labeled with these sera. No mature ones, and of course, gametes are very efficiently labeled. So this is the first quantitative evidence for the detection of signal on the red blood cell surface of stage 2 infected red blood cells. When you look at that from another angle, you can see that those sera that detect both stage 2 and asexual infected red blood cells, they have the highest titers. And these also increase with age. While those sera that only detect immature gametocytes have lower titers, and these increase much slower with age, probably reflecting the lower abundance of gametocytes compared to asexual stages during human infection. So here, this is just a field of view of live stage 2 and asexual parasites mixed here in red the gametocytes because we used the transgenic line, and you can see the green surface labeling, so these are intact cells. If you look at the stage-specific labeling, then most of the cells in the population of the asexuals are strongly labeled with human IgG, about 40% of the stage 2 gametocytes, and the stage 3 already are very weakly labeled. Stage 4 and 5 are basically not labeled. So that suggests that during gametocyte development, somehow the surface of the red blood cell is progressively remodeled and all the immunogenic antigens become absent, which is probably important for transmission and the circulation of the mature stage. So then we wanted to identify the antigens on the surface of the stage 2 gametocytes. We used a series of complementary approaches, initially basically producing a lot of stage 2 gametocytes and then either shaving them on the surface with trypsin chymotrypsin or keeping them intact. And then we generated so-called gametocyte ghost membranes, and first looked at them when we ran them on the gel and probed them with Malawi serum. So these are two sera that detect both stage 2 and asexual stages, and you can see this is a subset of trypsin-sensitive bands that you can see when you probe with both of these sera. So here, for example, and here. And here, and these seem to be actually present in the serum of both of these patients. So then next we performed a whole lane in-gel digestion and LCMS together with Roald Glazen's group, and we identified a total of 235 antigens with at least 1.25-fold enrichment in the minus trypsin sample. So these are shown here. A subset of 30 putative surface antigens were predicted based on their domain or signal sequence and the pexel or KNEP. So they are most likely exported into the host cell and maybe on the surface. 28 of those, so basically all of them, are expressed both in asexual and gametocyte stages. We also used these same two populations and infected 6 mice each, and then basically took the serum and ran the sera on the same protein array that Will Stone described before, and like that identified an additional 4 putative candidates that showed differential signal and were targets that were probably exported into the host cell. So in total we had 34 putative surface antigen candidates, 30 of those expressed in asexual and gametocyte stages, so shared antigens. We made antibodies against 9 of those, and again tested their surface exposure first by Western with plus-minus trypsin samples. You can see differences in some of them, but they're not very strong, probably because only a fraction of protein is on the surface and the rest is never getting exported. When you take live cells and do live microscopy or flow cytometry, you can see a differential effect in most of them, plus-minus trypsin, with the strongest effect actually here and here with these two proteins, GEX7 and 10, that previously Philippe Déter's group has demonstrated to be on the surface of the asexual infected red blood cell, and it's also expressed in gametocytes. So altogether, 6 out of these 9 antigens were validated on the surface of the infected red blood cell. So here you see these GEX7 and 10. They seem to be trypsin insensitive. There are 2 transmembrane domains with the extracellular loop. One of the key requirements for a malaria vaccine, of course, is that it can induce strain-transcendent immunity, and therefore that the target epitopes are conserved. So here we collaborated with Dan Eaves's group at Harvard with the idea to see whether we find conserved, a subset of conserved antigens amongst our candidates. So we looked at the population of parasites from Senegal and looked at non-synonymous nucleotide diversity and basically stratified the proteins from the array again in internal and secreted ones, and then made an arbitrary cutoff for for the level of IgG responses they mounted for low and high. And you can see only secreted antigens with IgG levels above a certain threshold seem to be immunogenic enough to be under immune selection, which of course is to be expected, but it's good to see. And there's a couple of candidates like Guest that have come up in other experiments as well. Another way to look at that is to see whether there's population differentiation or FST. And so we looked at the parasite genomes from these Senegalese parasites and compared them to Malawi parasites, and again stratified by intern and secreted antigens. And you can see a subset of our antigens are on the very high divergence or differentiation, and these are mostly mosquito antigens, as we could expect. So P47 is here, P230, P230P, because they're adapting to different mosquito populations. But the important thing is there's a number of red dots here and here, which are all our 34 candidates. Many of those seem to be relatively conserved. 8 of them of the 34 show low diversity and divergence, which for us would be one reason to prioritize them for further analysis. So one way to look at whether they could be functional is to see whether the IgG titers correlate with the level of with the parasite burden in the same patient. So here we looked again at these 3 cohorts that Will Stone presented and all the immune sera, and correlated the IgG titers across antigens with parasite burden from the same patients. You can see secreted asexual antigens are significantly negatively correlated with asexual parasite burden across these patients, while secreted shared antigens are slightly negative correlated with the fraction of gametocytes, so the proportion of gametocytes out of the total parasite load. If we look at our 8 candidates, 2 of them show no negative correlation for these 3 parameters. 3 of them show more or less negative correlation with the gametocyte fraction, and 3 of them show a negative correlation with both gametocyte burden and asexual parasite burden. So these are, of course, the most interesting ones. At least there is an association, but there may be actually a functional component to that these can at the same time reduce parasite burden and transmission. So one way that these could be functional is, as I mentioned at the beginning, through inducing phagocytosis. So James Beeson's lab has developed an assay using THP1 cells where you can expose infected red blood cells to human serum and then see how that induces phagocytosis of THP1 cells. So here we repeated that this is again the Malawi sera from that cohort with asexual cells, and as he has shown, you can significantly induce phagocytosis, but here we can show that this is also the case for gametocytes, and of course naive serum doesn't induce. Interestingly, the level of phagocytosis induction is positively correlated for asexuals and gametocytes stages with the serum, or with the intensity of the serum in flow cytometry assays. So altogether, we have identified anti-gametocyte-specific immune responses. They target antigens on the immature gametocyte-infected red blood cell, but not the mature ones. The levels increase with age. They recognize mostly shared antigens that are present on the asexual and the gametocyte surface, suggesting that they may have shared functions, maybe in the bone marrow, where we could target an interaction and therefore block development or release into the blood. The subset of these antigens is conserved and correlates negatively with gametocyte and asexual parasite burden, and the sera generally can induce both asexual and gametocyte infected blood cell phagocytosis. So now we have a short list of candidates which we would like to further investigate. We want to know what the function of these antigens is in protein or cellular interactions in the bone marrow environment. And of course, ultimately, we want to know whether these antigens can induce both strain and stage transcendent immunity, and whether therefore we can make a case for a combined blood stage and transmission blocking vaccine approach using a combination of these shared antigens. So with that, I would like to thank a couple of people, mostly Cassie Danzer, former PhD student in the lab, who has done most of that work. Of course, Tone Bosma, close collaborator, and Terry Taylor's group, and Miriam Laufer from the Malawi group, and a number of other people, including Curtis Hottenhauer and Diane Worth, and Dan Eavesy at Harvard, Phil Feldner, and Roland Ingleisen, and founding bodies. Thank you.
Yeah, hi, Sanjay, very nice work. Would you like to comment whether these stage 2 antigens you have identified could be these conduit of sequestration of gametocytes in bone marrow or elsewhere?
So the question is whether these antigens could be involved in sequestration in the bone marrow. That's your question, right?
Yes, yes.
Yeah. Well, that's what we hypothesize. So the three that I showed that are both conserved and they are negatively correlated with gametocyte burden and asexual burden, they are actually also conserved across the Plasmodium lineage. They're also present in Bergii, which for us is very exciting because it means we can also look in the mouse model, for example, with knockouts. So that's what we hypothesize, and that's what we are currently investigating.
I mean, it tells we should be paying a lot more attention to the early stage antigens, mostly taking stage 5 parasites from the culture and so on.
Yes, we should pay more attention, and I think they will be much easier as candidates also if we can just look at mature gametocyte burden as a readout rather than.
Why do you say that much easier?
Well, because what I mentioned at the beginning also is that the readout of such a vaccine approach would be the level of mature gametocytes in the blood. Yeah. Yeah. Oh, Colin, thank you.
Hi, Matt. Very nice talk and very exciting work. It's clear that with the power of the new tools, you're able to dissect things very nicely. But my question is actually similar to the previous questioner, which is what is going on in the bone marrow, and why might the parasites want to have these proteins on the surface of those early stage gametocytes, given that we don't expect there to be an absolute requirement for adhesive interactions in the same way that we expect for the asexuals. So I'm sure you've had some philosophical late night discussions with colleagues about what might be going on. Can you share any of your more radical
the
views?
early or the late night discussions? Well, so in general, I think the interesting aspect is that they are shared. If they would be gametocyte specific, I would be concerned about antibody titers, because gametocytes only make 1% of the asexual parasite level, and therefore the titers would be very low. But because they are shared, I think they have a shared function, and we see both gametocytes and asexual stages, for example, bound to the sinusoidal endothelium in the bone marrow and in the mice also in the spleen. And you could imagine that such an interaction can be blocked. And that's also suggested by the experiments that I showed from Pietro Alano. So I think the exciting part is that this is not a gametocyte-specific process, but it's a general process of parasite sequestration in the marrow that we don't understand, but that probably is required for both asexuals and gametocytes, and therefore the responses, of course, are triggered and driven by those against asexuals, and the gametocyte response is basically piggybacking on that one. That's the advantage. But what exactly that interaction is, we don't know.
Thanks.
Hey, Matt. Nice presentation. So, extending the previous question, have you looked at your shared antigens, and that are expressed in both asexuals and the stage 2, to see when those antigens are expressed in the asexuals? Meaning, could one of the reasons between the shared antigens be that these are myeloid antigens? And as you know, a lot of the myeloid antigens, like ROP3s and dense granules, those antigens end up on the surface of the parasite. And so is that the reason why you see those shared antigens between the early gametocytes and the infected cells?
I don't think so. So, for example, GEX 7 and 10 have previously been identified by Philippe Déter. They seem to be a chemokine receptor mimic, And they are expressed quite strongly on the surface of trophozoites and schizons in asexual stages, and they are also expressed in stage 1 and early stage 2 gametocytes in our experiment. So I don't think that there is myristoyl antigens in that list that are subsequently deposited on the infected red blood cell surface from stage 2.
Hi, thanks. Very nice. And so can you speak a little bit more about why only 10% of the twos? Is that
10%?
seem to have these on their surface?
Oh, yes, so that's a good question. So it's actually much more. The problem is that except for 3D7, all the strains have very significant non-specific IgG labeling, and then there's a lot of background. So that we have to subtract that. And the other thing is that even asexual stages, when we look in a population, there's probably 50 or 60% that are positive with any serum, human serum. So it's less than that, but it's anyway never 100%. So I think that's the last one, and then we have to move on. Well, you can.
I'm the last one. Matt, I'm interested in the proteins that did not have a PEX or a PNET-like domain. So you said that you detected by mass spec approximately 200 proteins, of which 30 looked like they fulfilled that criteria. So what did the other subset look like?
You mean the 200 that didn't have a signature that they are exported?
But were detected on the surface of the gametocyte?
Well, whether they have been detected by a differential shaving approach, that's the only thing we can say. So the shaving itself, of course, damages the membrane, and you always have noise. So I think a large part of that is just noise from the partial lysis of the red blood cell with the shaving itself. So I wouldn't pay too much attention to those, honestly.
Okay.
Okay.